Okay, so welcome to the third lecture in this Extensions of Mendelian Genetics series. This lecture is going to cover quite a lot of the chapter. Let's talk about sex for a lot of this lecture. We're going to talk about sex chromosomes a little bit, different types of sex-related traits, sex-influenced and sex-limited traits. And then we'll finish by talking about a couple of different types of single gene problems, lethal alleles that occur, and then pleiotropy. And again, we are talking throughout this entire lecture about different ways that single genes can have different effects when it comes to phenotype. So let's talk about sex chromosomes to begin with. It is true that many species have males and females, and that those males and females differ genetically in their sex chromosome composition. You guys all know about this for human beings, right? Girls are XX when it comes to the composition of their sex chromosomes, and males are XY. There are certain traits that are governed by genes on these chromosomes, as you guys are also aware. You should also know that there are genes on the X chromosome in particular that are essentially used for things not related to sex determination, because all human beings have at least one X chromosome. But males do only have one, they do not have two. And this leads to some genetic problems whereby there are things called X-linked diseases that only affect males in the population, and mothers are carriers. Now think about that for a second, and it will make some sense to you, because what you guys know from some of the other stuff we've looked at, right, is that you have dominant and recessive alleles, like in the case of a plant height, for example. If you have at least one capital T, you're tall. If you're homozygous little t, you are short. You're only short when you don't have one big T, right? Now, males only actually have one version of the X chromosome. So if there's a disease on the X chromosome, which is acting like a recessive disease, in other words, if you've got a single dominant allele, you're fine. Well, males only have one copy of all of the genes on the X chromosome. They're not heterozygous for those genes because they don't have any other copies. They are what is called hemizygous, those genes. But genetically speaking, right, if you inherit just one recessive allele from your mum when you are conceived, you don't have a wild type copy to rescue it, and therefore you come down with what's called a sex-linked disease. So, for example, Duchenne muscular dystrophy is an example of a sex-linked disease. So what you see is that if you look at a female up here, she is unaffected phenotypically, but she's a heterozygote. She has one disease copy on one of her X chromosomes, but she also has one good copy. So therefore, she looks normal, right? Classic Mendelian situation like big T, little t. But when she has kids with this man, the boys that are created in this family, of which there is just one right here. So this couple, one boy, had one boy, and then 2, 3 girls. The boy has Duchenne muscular dystrophy because he inherited the X chromosome from his mum, the Y chromosome from his dad, and it just so happened that the X he got from his mum had the mutation on it. So he shows the disease. Now, she went on to have 3 daughters, each of which have 2 X chromosomes. One came from her and one came from him. 2 of these daughters inherited the mutation allele, or the affected allele from mom. They are heterozygotes because they inherited a good one from dad. And then this daughter inherited a good copy of the X from mom and a good copy from dad. None of these show the disease because they have at least one good copy of the gene. But these two are carriers. Therefore, when this woman had a kid with this unaffected male, one of their kids who's a male, the males, remember, have to have one X from their dad and one of these two X's from mom. The male is affected. With this daughter that had kids with this unaffected man, she actually has a son that's unaffected, so he must have got one X chromosome, and it must have been a good one from his mom, and the Y chromosome from his dad. Therefore, he is a boy who is unaffected, etc., etc., etc. So if you look at that, oh yeah, so here's one example of a real human pedigree, hemophilia, which is a X-linked trait. And Queen Victoria in the British royal family was a carrier. And if you look at this, pedigree of the British royal family, you see that Queen Victoria was a carrier, and then there was lots of male hemophiliacs that ran throughout this royal family. None of which, interestingly enough, are seen in the British royal family. So here's Princess Anne, Prince Charles, Prince Andrew, and Prince Edward. But there's a lot in the other branches of this family, which went on to be part of the German royal family, and the Russian royal family, and the Spanish royal. Let's take a quick look at how Punnett squares work when you're looking at X-linked traits. You have to be a little careful with them. And they're interesting because what you'll see is that Punnett squares of X-linked traits show different results depending on which sex is affected. So we're going to look at some examples of when you transmit diseases via the X chromosome. So here's an example of that occurring. Here is an unaffected female and an affected male carrying a disease allele on his X chromosome. She has two normal X chromosomes. So in this case, she has two X's, both with a capital D. This is Duchenne muscular dystrophy. Capital D being the dominant version of this allele. Here is an affected male who has muscular dystrophy. He has the little version of the dystrophy gene, and he has a Y chromosome because he's a male. So when you think about the possible gametes that she can make, you put them on a Punnett square, there's only one possibility, X big D. So here are the two possible gametes from the female. When you think about the gametes this guy can make, this male, he can either make gametes with an X or with a Y. So here are his two possible gametes from his sex chromosomes. And then if we fill in the Punnett square, we see that in every instance, there is an X capital D, which means that whether or not you're a girl or a boy, you're going to be unaffected. And that's because all the females will definitely get one mutant X chromosome from dad, because the only way they can be female is to have two X chromosomes, right? One from dad and one from mom. But both of the possibilities from mom contain the dominant wild type version of this allele. Therefore, they're unaffected by the disease, but they're both carriers of that disease. If you look at the boys that come out of this progeny, any boys have to inherit Y from dad, the only Y that's available, and one X from mom. Both of these males will be unaffected. So that's what you get if you cross an affected male with an unaffected female. If you do what is called a reciprocal cross, shown on the right here, reciprocal in this case just means, in this case, the genotypes and the phenotypes are flipped. So now you have an affected female, homozygous recessive, with an unaffected male. So the opposite of the genotypes over here, or the phenotypes, I should say, over here. When you have that situation, you'll see that there's differences in the sexes. So again, the sperm have to be either XD as one gamete or Y the other gamete. And the female gametes are both X little d. And if we fill out our Punnett square, you'll see that both of the females are unaffected and carriers. Both of the males, because they have to get one X from mom, are both affected with muscular dystrophy. And so you get this very specific difference in phenotypes depending on who the parents are. So that's how X-linked diseases work. Now, there are certain traits that are influenced by sex, but not sex-linked. What does that mean? What it means is sex-influenced traits have this weird situation where an allele is dominant in one sex, but recessive in the opposite sex. These alleles, these genes are present not on the sex chromosomes, but on the autosomes, which is every chromosome that's not sex chromosome. They are not sex-linked traits, but you do see differences between the sexes. Because we're talking about an allele that acts dominantly in one sex, but recessively in the opposite sex, the only time you see these is in heterozygotes. Let me show you what that means, because it's way easier to understand. So one example of this type of trait is scurs in cattle. So scurs are these, if you look at this picture of this cow down here, you see, or this bull, I should say, down here, I guess. It has scurs, which are these little horn-like growths on the frontal bone of the skull. Now, this is a sex-influenced trait. What does that mean? Well, there's one gene involved, represented by SC for scurs. So this is not two genes. It's confusing, I know. Genetics is like that. This SC means scurs, the gene. Capital S little c is dominant, little s little c is recessive. So there's three possible combinations, right? Homozygous dominant up here, heterozygous, or homozygous recessive. Okay, now if you look at the phenotypes in males, what you see is that homozygous dominant, scurs, heterozygous dominant, also scurs, so sort of classic Mendelian situation. Homozygote recessive, no scurs. So again, classic big T, little t type Mendelian situation, right? Now look at the phenotype in females. Scurs in the homozygous dominant animal, and that's normal. No scurs in the homozygote recessive animal, again, you would expect. But interestingly, look at the heterozygous phenotype. Even though there's a dominant version of this gene, it looks like it's a homozygote recessive, but only in the heterozygote situation. So these two heterozygotes look different in males and females, even though genotypically they're the same. That's an example of a sex-influenced trait. So last but not least, when we're talking about traits that are dependent on one's sex, there's something called sex-limited traits, which are basically traits that only exist in one of the two sexes. These are normally traits that are controlled by some aspect of what makes you a boy or a girl. So normally sex hormones or something in the sexual development pathway. An example of that is, sex organs. So those are sex-limited traits. In humans, ovary development is limited to females. Testes development is limited to males. That's because of the sexual development pathway. There's also things like, strong facial hair. That's also limited mostly by sex. That's due to sex hormones. In birds, as you guys probably know, like so this example of chickens, the males often have extravagant, more ornate plumage. Females, that is an example of a sex-limited trait. Again, that's not sex-linked, it's just limited by sex. Okay, so last but not least, there's two other phenomena, genetic phenomena that you need to know about. One is lethal alleles that we haven't really talked about yet, and the other is CLIAtrophy. So lethal alleles are pretty simple to understand. It's basically an allele that kills the organism. Normally, they act recessively. They're normally loss of function recessive alleles. And they're normally the result of mutations in what are called essential genes. What is an essential gene? An essential gene is simply one that's required for you to live. So one example of that would be DNA polymerase 1, right? Or DNA polymerase 3. So DNA polymerase 3, as you should remember, is the protein that extends DNA during DNA replication, right? If you don't have that, you can't replicate your DNA, you're dead. That's an example of an essential gene. So a lethal allele is one that can potentially cause the death of an organism, normally in a homozygous recessive situation. Normally, if you have one working copy of a gene, it is sufficient to allow you to live. Interestingly, about 30% of the genes that you have thought to be essential for survival. 70 are not. Kind of a crazy number. There's a few different types of lethal alleles that are interesting. One is called conditional lethal alleles. Those types of alleles, we actually need to hold them, italicize that one too. So conditional lethal alleles, just like it sounds, are lethal under certain conditions. For example, there's lethal temperature sensitive mutants in Drosophila. So they are mutants that normal temperature or at one temperature, they're fine. You change the temperature, those alleles then become lethal to the organism. A bit like the Arctic fox we talked about in the other lecture, but this time instead of changing the coat color, it means that death occurs when you change temperature. Semi-lethal alleles kill some individuals of the population, but not all of them. An example of that is Manx cats, which we'll look at in just a second. Why could alleles be semi-lethal in some cases? Sometimes environmental factors and other genes can prevent them becoming completely lethal. It just depends on the gene and the allele. But lethal alleles themselves and semi-lethal alleles can cause ratios that seem to deviate from Mendel in some way. So here's the example of Manx cats. Manx cats actually carry a dominant mutation. We're going to notate that as M here, that affects spinal development in those cats. Manx cats specifically look like this. They have short tails. This actually, it can have a different amount of expressivity. The tails can be slightly longer or much longer than those shown here, which are effectively no tails. Interestingly, if this mutation that is dominant and affects spinal development is present in two copies, in other words, if you're a capital M homozygote, then, as is not really that surprising if you're talking about spine development, if your spine development is extremely messed up, then you die early in development. Now, what does that mean when you talk about Mendelian ratios? So this Punnett square is like, a million that you've already drawn. Two Manx cats, which are heterozygous for this dominant mutation. You cross them together, you get a classic Mendelian genotypic ratio. So, two hets, right? You're going to get homozygous dominant, homozygous recessive, and two heterozygotes. But look at the phenotypes, the phenotypic ratio that you get. You get two of these four, which are heterozygotes. Those are going to be Manx cats because they'll have messed up spines and therefore shortened tails. If you have homozygote recessive, then you don't have any copies of this capital M, and you look like a normal kitty cat. But if you are homozygous dominant, you actually die super early in development. You're never actually made as an individual, so you just don't exist. So then the phenotypic ratio is 1 to 2 here, 1 non-Manx to 2 Manx ratios. Notice that Mendel's laws are still perfectly fine here. You're still getting independent assortment, and you're still getting segregation, but the phenotypic ratios are different because of, phenotypes that exist, one of them being lethal. Last but not least, we've got to talk about CLIAtrophy. This is an important genetic concept. And this is the idea that a lot of genes in your body actually have multiple biological effects, not just one type of biological effect. Now, the fact that one gene can have multiple phenotypic effects is known as CLIAtrophy. And this can occur for many different reasons. So the gene product of any given gene, or the protein, right, that gene makes, can affect cellular function in more than one way. It can be expressed in different cell types, the gene. And the gene can be expressed at different stages of development. And a couple of examples of these kind of things are cystic fibrosis, sickle cell anemia. So sickle cell anemia, for example, that you guys know about, right? If you have that phenotype, if you have sickled cells, then your blood is filled with those sickle-celled red blood cells. Now, the phenotypic effects of that just simply depend on where problems arise. So problems arise for sickle cell people where those sickle-celled red blood cells clog capillaries. And that can happen anywhere in the body. So if you have a sickle cell problem, you could have a cell blockage, a capillary blockage in your arm, or your brain, or your liver. And whenever that happens, you're going to have a different phenotype, a different outcome, right? That is a pleiotropic disease. Cystic fibrosis is a disease that's based on one gene, and it actually messes up a sodium transporter that transports ions across a membrane. That gene is expressed in most of the cells in the body, but it does different things in most of the cells in the body. And because of that, you have lots of different phenotypes if you have cystic fibrosis. One gene product that does different jobs in different tissues, therefore you see lots of different biological effects. And that is, sorry about that, is all I have to say about that particular so we're done now with third installment of Extensions to Mendelian Genetics. And the next one, we're going to talk about gene interactions, which is a complicated topic. So that's why just one part of the chapter for a whole lecture. All right. See you for that one.